Yeah. Hi, everybody. Good afternoon. I want to thank the meeting organizers for giving me this wonderful opportunity to talk a little bit about my work. Hopefully, you will find it somewhat interesting. Anyway, so in our lab, we study Staphylococcus aureus. Pretty sure I don't need to convince this audience that Staph is a pretty prominent pathogen. And in particular, what kind of makes Staph such a prominent pathogen is that it has a really good ability to adapt to different environmental stresses, such as those encountered within the host. One of those stresses that Staph and other pathogenic bacteria face during infection is a limitation of metal ions, such as iron. And this limitation is actually on purpose and mediated by a host using a wide variety of different metal acquisition systems. And it's all this to sort of prevent bacterial proliferation. An example I listed here is that it uses proteins like transferrin and lactoferrin to kind of bind free and available iron. But you can see it is sort of complicated, so the host goes to great lengths to limit bacteria from these essential metal ions, including particular iron. So since the host does this, bacteria need to be able to respond and adapt to this iron limitation. And in the canonical model for iron regulation in bacteria, they use a protein called FER, or the ferric uptake regulator. And if you're not familiar with FER, how it acts is pretty simple. It's when during iron replete conditions, FER gets metallated, and by being metallated, it's able to bind to a DNA sequence known as a FER box. FER box are typically located in the operator region of target genes. Therefore, FER binding typically leads to transcriptional repression. But during iron-limited conditions, FER releases the DNA and transcription of the target genes are allowed to occur. These target genes are now going to be upregulated. And some of these target genes include various iron uptake systems that you can imagine are important to be, or need to be going to be expressed in the absence of iron. And this model also shows that there's another protein here that kind of plays a role, but kind of the main character here is FER. So we know a lot about how Staph aureus uses this mechanism to upregulate iron acquisition systems, and we really know that a lot about this different iron acquisition system. But what we don't really know is that how Staph aureus and FER regulates iron usage in particular in response to iron limitation. So before I joined the lab, Jeff was sort of really interested in iron sulfur clusters, and he kind of wondered whether FER had any impact or regulatory effect on iron sulfur cluster biogenesis. So he looked at a familiar iron sulfur cluster using enzyme in aconitase, which is typically familiar since it's the second enzyme in the TCA cycle. So in the essence of this, we looked at the aconitase activity of a wild type. Oh, it's look a little blurry. Anyway, we looked at the aconitase activity of a wild type versus a FER mutant. And we saw that in the absence of FER, we had basically zero aconitase activity. And this could be complemented. And this phenotype could also be kind of copied in the absence of iron. So we cultured the cells in iron-rich and iron-limited conditions as done by adding a metal chelator called 2,2-dipyridol, as you can see here. And if you culture the cells during iron-limited conditions, then aconitase activity also goes down. So where previously I told you that FER normally acts as a repressor during iron replete conditions, FER seems to be acting as sort of like an activator of aconitase activity, meaning that in the absence of FER, we have an inactive TCA cycle. And this kind of was really interesting to us. And because FER lacked aconitase activity, we also saw that a Staph aureus FER mutant had a growth defect when cultured under defined media where amino acids were sole carbon sources. This would be expected since amino acids sometimes enter metabolism through the TCA cycle. So lack of aconitase or TCA cycle activity would lead to this growth phenotype. And we were really interested in trying to find how FIR was impacting this TCA cycle activity, we kind of wondered whether or not there might be like an intermediary. And since we had our mutant with a clear growth phenotype, we decided that maybe we could find this intermediary regulator using a suppressor screen. So suppressor screens are pretty simple, but they can be powerful. And in essence, how they work is that we have our initial mutation, which leads to growth phenotype, in this case, basically no growth under this medium. But what we did was we were able to isolate strains that contained the original FIR mutation, but had a second site mutation somewhere else in the chromosome that restored the original phenotype and allowed it to have restored growth in this media with only amino acids, therefore kind of suppressing the effect of the original mutation. So we were able to find these FIR suppressor strains. As you can see, FIR doesn't grow on amino acid media, but these FIR suppressors had increased growth in this media. And once we assayed aconitase activity, we saw that these FIR suppressors also had restored aconitase activity. So like signaling to us that this mutated gene, at this point, we didn't know what it was impacting the regulation between FIR and aconitase activity. Maybe it was somehow mediating the repression of the TCA cycle that we've been seeing. So to try to identify and find out what this gene does, we sequenced these like FIR suppressors and found that all the FIR suppressors that we isolated all had the exact same mutation in the same exact gene, which codes for a non-coding small RNA. But before we could get all excited, we first had to make a deletion mutant. So we know that a FIR can grow during amino acid media. But once we took away the IsrR allele, then we saw that we could restore the growth of the FIR mutant. And we could also add back IsrR, showing that we could revert back to the original phenotype. All of this hoping to show that indeed it was this gene IsrR that's mediating this growth phenotype that we're seeing here. We also determine aconitase activity. As you can see, we already know that FIR mutant lacks any aconitase activity, but if you take away IsrR, then we see like a partial restoration of aconitase activity. All this suggesting that maybe this IsrR gene, when expressed in the absence of FIR, was somehow targeting the TCA cycle. Additionally, I don't show it here, but we do show that IsrR is indeed regulated by FIR. So if you don't have FIR, then this gene is going to be expressed. So we were interested in following up how exactly this small RNA was impacting the aconitase activity in the TCA cycle. So naturally, we decided to look at the sequence of our small RNA, IsrR, and we compared it to the sequence of the aconitase transcript, because we know that small RNAs act at a transcript level. So you can see this predicted interaction was very interesting to us, because the predicted interaction wasn't just anywhere randomly in the gene, it actually overlapped the aconitase ribosomal binding site, which if you sort of try to picture it in your head, if you have your ribosomal binding site and something is now binding to it and sort of like protecting it, then that would sort of like impede the ribosome from assembling, and that would lead to translational repression. So we wanted to see whether or not IsrR was impacting the translation of the aconitase transcript. So what we did was a really simple technique where we took a constitutive promoter and we cloned in the 5' UTR of aconitase, which sort of included this region over here, and also some the starting codons of aconitase, and we fused that to GFP so that we can measure fluorescence as a signal for aconitase translation. And as you can see here, it's sort of like blurry, but a FIR mutant has decreased aconitase translation, which is expected since we already know it has decreased aconitase activity. But now we see it also has decreased translation. And this is recovered on the FIR IsrR double mutant, sort of like signaling to us that the decrease in translation that we see in the FIR mutant was due and dependent on the presence of this small RNA. So we naturally assume, well, this sort of like to validate that it impacts the translation by directly binding to it, we did what are known as EMSAs, or electrophoretic mobility gel ships, which is a lot of complicated but simple to explain, where you normally have your small RNA, typically small, so it's going to migrate to the bottom of the gel. If you add the aconitase transcript and IsrR is binding to the transcript, then normally it's gonna, that complex is gonna have a higher molecular weight, therefore it's gonna cause a shift in the band, and this is exactly what we see over here. So now we're trying to, with this knowledge, we can put together a couple more pieces of the puzzle and know that during iron limiting conditions, FIR acts as a repressor of IsrR, which I didn't show, but it does. But in the absence of FIR, IsrR is expressed, it binds to the aconitase transcript, leads to translational, so like repression or inhibition, and that's what's causing the inactive TCA cycle that we see in the absence of FIR. So we were really excited at this point, but aconitase is just one gene, so we wondered whether or not IsrR was impacting the expression of other genes that are in the TCA cycle. So we measured the activity of genes like succinate dehydrogenase and malonyl reductase, just like these different steps, and show that it has similar aconitase type of regulation for these two genes, and it also directly bound their RNA transcripts. So hopefully the work I presented here can sort of like convince you to this model where upon iron replete conditions, FIR acts as a repressor, as we kind of know it does, of IsrR, but during iron limiting conditions, FIR repression is relieved, we get expression of the small RNA IsrR, it binds to these different TCA cycle mRNAs and leads to their translational repression, and that's why, and all that leading to an inactive TCA cycle. And sort of like why this matters, or kind of like the relevance of this work, is that in the TCA cycle, we have genes like aconitase and SDH that use iron as a cofactor. Therefore, what we hypothesize is happening is that during iron limiting conditions, we sort of like are shutting down the expression of these genes that use up iron, sort of like to decrease iron usage in the cell. And that would be a way to sort of like spare the maybe the limited amount of iron that there's left for the more essential processes. So this sort of like gives us an insight into how Staph adapts to iron limitations, which can be encountered in the host or maybe in the environment, as stated in the morning talk, and kind of provides an insight of how what is happening in the adaptability mechanisms that Staph uses to overcome this limitation. So probably running out of time, but I wanted to also kind of share that with perform mouse models, and we infected using a model of acute pneumonia. We infected it with our different strains, and we saw that if you delete IsrR, we saw that they had attenuated virulence, meaning that they had like decreased colonization in the lung. And we can talk more about in the poster if you want. I'm poster number 2, but I've also shown that IsrR impacts other cellular processes that are associated with iron, not just the TCA cycle. And with that, I want to thank my advisor, Jeff, and the lab members, our wonderful collaborators, and finally the funding solutions. With that, I'll take any questions.
And IsrR, I'm thinking about, we have FIR that's sending iron. Is it that IsrR is like expanding glycogen, or are there meat regulators additionally to FIR that regulate IsrR expression? I'm just trying to can you differentiate between why would you need both of these systems or something?
So what you're saying is basically, why can't FIR be a direct like activator instead of using this? Well, using small RNAs is pretty helpful and pretty useful, and bacteria typically use small RNAs for stress responses since they don't really need to be translated into proteins. So I feel like using a small RNA gives you an advantage in managing these different stress responses. So it's like you don't need to translate it just by transcription, and it also impacts at translational level, whereas protein level. So I feel like it's like using a small RNA gives like an advantage to manage like stress responses.
Thanks, Alex. What would be a more essential function for iron in cells as DCA cell?
Well, I would argue that like it's been like studied that some iron-using proteins are also involved in things like DNA replication or DNA repair mechanisms, which I would think would be more essential than just regular TCA cycle. But it's an interesting question. I guess it'll be good. I guess we could somehow maybe find a way to like track where maybe the iron is going, but that's something we haven't tested in the lab, but it's a definitely good question.
Great thought. So I was wondering, since there's also enzymes in the TCA cycle that use metals like magnesium as cofactors, would you expect magnesium low conditions to affect the TCA cycle as well? Or is the system of iron regulation in Staph specifically related evolution to adapt to the host removing iron?
Well, I'm unfamiliar as to we see a similar type response to magnesium, but that's a great question. I do know that, yeah, I'm not really sure about magnesium, but there's similar type responses in when it comes to like manganese that use Staph also has like a manganese small RNA regulator. But in terms of magnesium, I haven't really looked into, I haven't really read anything regarding that, but I guess it will be interesting to look at.